Morning. It's my pleasure to introduce a friend and colleague of mine, Zhang, who is from Hackensack School of Medicine. Yi received his BS from Zhang Medical College in China, and his master's at Suzhou, which I visited several times. It's a great place. And then received his MD PhD at the University of Tokyo School of Medicine, after which he became a resident and fellow in pediatrics, again in Suzhou, and then did postdocs with Jim Wilson, whom really has pioneered a lot of the adenovirus work at University of Pennsylvania, and Steve Emerson, who later became head of Haverford College, president of Haverford College, but he's done a lot of seminal work on T-cell responses. He's been appointed as associate professor in the Department of Immunology at Suzhou, and then became an assistant professor at University of Michigan, recruited to be an associate professor and full tenured professor at Temple. And he's now professor and director of the Center for Discovery and Innovation at Hackensack. He's had a number of awards. I'll name a few: Special Prize for Scientists with Extraordinary Academic Achievements, Damon Runyon Award, a CS Research Scholar, DoD New Investigator, and a DoD New Idea Awards. He's a frequent reviewer for many foundations and NIH study sections, and was a full member of one study section, TTT, Transplantation and Tumor Immunology Study Section. He's an associate editor of Journal of Immunology Research and Therapy, and he's organized symposiums on epigenetics. He's supported by 2 R01s as PIs, 2 R01s as co-investigator, SPORE grant, and he's a co-leader of a program project, co-leaders of this project on the SPORE grant. So he's had 3 areas of productivity over his career. He began with studies in dendritic cells, differentiation from stem cells, examined the role of TGF-beta and other cytokines, and then exploited antigen-presenting cells in the liver to look at recruited and activated allogeneic CD8 positive T cells, which are rapidly primed in vivo, and made a really important discovery, which was a discovery of, first discovery of memory stem cells, was from Yi in a graft versus host disease model with Steve Emerson. Yi's done seminal work in delta-like ligand support by dendritic cells and other populations, and has then spent more recently time on epigenetics and the identification of stem cell transcriptional programs in alloactivated CD8 cells, effective histone methylation inhibitors, inhibition of histone methylations on ongoing GVHD responses, and then is focused on EZH2 as a critical epigenetic regulator of allogeneic T-cell responses. He's examined EZH2 phosphorylation and maintaining CD8 memory T cells for anti-tumor work, and these have been reported in high-impact journals. So my pleasure to invite you to give your talk on epigenetic regulation of T-cell alloimmunity tumor immunity.
Thank you. Thank you, Dr. Blazer. It's my great honor to be here. So in particular with our pioneer scientist and physician scientist, Dr. Blazer, in the field. So just to share one thing with you, Dr. Blazer is like a kind of BMT GVH biology library. You know, whenever you have a question, check his review paper, his paper, you find a solution and you move on. And you will see from my discussion today, how I learned this, how I use that. Thank you, Bruce. So today I will discuss about the epigenetic regulation of T-cell alloimmunity and autoimmunity and tumor immunity. So this discussion includes 3 parts. One is T-cell EZH2 and the GVHD. And the second part will be EZH2 modulation, calcium signals in the T-cell. And the last part to switch the gear, to be more about tumor immunology, how easy to incorporate the T cell proliferation and senescence, therefore maintain the proliferative capacity and the persistent capacity of a CAR T cell for the infectious therapeutic therapies. So we all know that we know here is an example from my postdoc time even I learned from Bruce Blazer's paper about the GVHD pathology. So it starts from the preparative conditioning. You have activated antigen presenting cells, including dendritic cells, to produce this kind of molecule, including notch ligand, and activate the T cell, including naive CD8 and CD4 T cells. And these cells are producing inflammatory cytokine, cytotoxic molecule, and then induce a tissue damage. So the most significant and also the determined factor is the damage to the GI, the intestine system, because that is life-threatening. So, and then you have a GVHD, patient may die. So the tissue, inflamed tissue may produce some suppressive molecule, try to calm down this inflammatory T cell response, excuse me. But on the other hand, they can serve as a kind of place to amplify, propagate the T cell response. Therefore, this kind of a cascade reaction caused the severe GVHD. And in the field, existing approach basically using the pan-immunosuppressive agents to suppress the immune response. But we ask a question whether you can selectively reduce the tissue inflammation, therefore you can avoid, ideally, the risk of infection and the immune deficiency after the pan-immunosuppressive agent therapy. Bearing this question in mind, so we and many other groups together, work on understanding how the local tissue induce the T cell response, sustain the T cell response. For example, the host non-antigen, non-hematopoietic tissues, such as intestine epithelial cells, stem cells, fibroblasts, they contribute to the expansion and maintenance of the alloreactive T cell in the local tissue, in particularly intestine epithelial cells. They express a high level MHC class 1-2 molecule that produce chemokines and other cytokine, and then they can recruit and expand the alloreactive T cells. The hematopoietic tissues, such as the dendritic cells, macrophage, and the MDSC, all play a role in the modulation or activating and sustaining the alloreactive T cell response. And these molecules play also important role, including the antigen-presenting molecule, notch ligands, and the cytokine. So some group also spend made quite efforts on characterizing the tissue infiltrating cells, including the identification of tissue-resident memory-like phenotype cells, TCF1 positive progenitor-like cells. One of the work is published in Immunity by Warren Schrammcheck's group. He's from Pittsburgh. He identified this cell population is important for sustaining the alloreactive T cell in the GI and other GVHD tissues. These progenitor-like or memory-like cells are maintained by transcriptional program, but exactly the precise role of this epigenetic regulator in regulating the tissue infiltrating cells remains largely unknown. So this is a wider open field for us to study, to understand what's going on there. Therefore, you can develop the approach to selectively reduce the GVHD in the local tissue. Therefore, you can avoid the use of the pan-immunosuppressive agent. So this is a paper Bruce mentioned about, it's 20 years so published Nature Medicine about the discovery of mammary stem cells. At the beginning, people did not really were taking this kind of idea. So we had a lot of trouble, for applying funding and so on. So, at that time I moved to Michigan. So my question is, how can I survive if people, did not take the idea? So we started working on the molecule pathway. So if you have a stem cell glycogen program, regulating these alloreactive T cell, and then we can prove the concept, say you need a stem cell gene program, and you do have a stem cell population there. So we did RNA-seq. This is a unique mining energy mismatched GVHD mouse model. So we discovered 39 epigenetic regulator. Among them, EZH2 is the top molecule highly expressed in the alloreactive effect cells and also stem cell memory T cells. So EZH2 is a histomethyltransferase, a form a complex with other molecule. One is a Suis12 EED. So these molecules EED bind to the DNA. So EZH2 itself does not directly bind to DNA, but in the context of this complex, EZH2 now bind to the DNA. And the primary role of EZH2 is silencing gene expression, but it has a non-chronic function, and I will discuss more about that today. So this is the paper after I published, after I became independent and set up my own lab. Dr. Blazer was the editor at that time. He emailed me, saying, you open a new window and you can look back at these many molecules. We actually found 401 stem cell relevant genes there. So so far over the past 15 years, I basically focus on the EZH2 because we believe this is a very important molecule, play a fundamental role in the regulation T cell immune response. So one of my postdocs, Zhang, she crossed over the conditional knockout mice to the CD4 established the T cell specific knockout EZH2, and induced the GVHD. And she found that if you knock out the T cell EZH2 in mature T cell, you don't cause such GVHD now. And one of the major cellular molecular mechanism is that if you don't have EZH2, now T cell cannot expand. They can proliferate, they can differentiate into effect, but they cannot persist. So survival is a major issue of these T cell lacking the EZH2. And so we ask a question: how can we translate this into the clinical application? So at that time, there was one EZH2 inhibitor called DZNAP, naprotein, which selectively reduced the histomethylation and induced the EZH2 depletion. If you gave the EZH2 to the mice even 7 days or 10 days after bone marrow transplantation, GVHD was ongoing at that time, you can still arrest the GVHD, and very powerful. We have a data also not published. You can use a DZNAP to prevent the hot allocardial graft rejection, and you can use the torrents almost forever. But unfortunately, DZNAP is not available for clinical application. That was initially developed in NIH. And for inhibiting the Ebola virus, but somehow it caused when we used it for Ebola virus, inhibition caused kind of immunosuppression, so it's stopped. And then patent expired, and no farmer want to develop this drug. So we moved to the next generation of EZH2 inhibitor, that is tisomitostate and UNC199. So these two molecules selectively reduce the H3K27, which is characterized by EZH2, but it does not significantly deplete EZH2 protein, in particular in vivo. So if you gave these compounds to the GVHD mice, none of them work. So one of my PhD students at that time spent like nearly 1.5 or 2 years to work out and what's going on there. And she finally got the results like this. She cried. So it's a really kind of frustrating at that time. And then we revisited the EZH knockout cells. So for EZH knockout cells, you have a protein, EZH protein totally depleted. But for the drug treatment, in particularly if you gave the teasomidal state in the GVHD mice, you don't eliminate EZH2 protein. You even like a relatively increased EZH2 protein expression if you gave a higher dose. So we ask, say, would the EZH protein be important for alloreactive T cell response? If yes, how can you induce the depletion of EZH protein? So we did the mass spectrum analysis, and we found the EZH actually interact with the Hitchcock protein HSP90. So when we use HSP90 inhibitor AUY922, which is developed by I think a GSK for the tumor therapy, we found the EZ2 now you depleted, and EZ2 HSP90 really interact each other. We use a core IP to verify that. Now you see the reduction of the GVHD, and we tried testing several other models. So this indicates that you need to deplete EZH2 in alloreactive T cells in order to arrest the GVHD. And so again, Dr. Blazer's group did a great job to make the progress. His group studied the EZH inhibitor combined with the JQ1, the BET domain inhibitor. This combination can suppress the chronic GVHD, and his group also made significant contribution to understanding how easy to modulate the alloreactive T cell response, in particular in the context of chronic GVHD. So he found that if you inhibit EZH2 using this JQ5, you can reduce mTOR and c-MYC. In a separate study shown by other group using the cancer cells, actually easy to interact with the c-MYC. So this finding published in Blood, which indicate that EZH2 inhibitor in combination with the JAK1 BET domain inhibitor can be used for suppressing the chronic GVHD, which is a major issue still in clinic. So we're still not satisfied with the EZH2 inhibitor because you may have the risk of inducing like a pan immunosuppression in vivo because you induce a systemic suppression of EZH2 in T cell, given the importance of EZH2 in promoting the survival expansion of alloreactive T cell and maybe some other tumor active T cell and so on. So we ask, say, could be possible downstream molecule pathway of EZH2 may be a target for you to selectively reduce the tissue GVHD. So we identify ID3. ID3 is a downstream molecule of EZH2. If you knock out EZH2, you see reduce the ID3 expression. And we did the cut and run in a chip assay, and we found that easy to direct bind to the ID3. Of course, easy to also suppress other molecules such as ID2, EOMers, and so on. So ID3 is a DNA binding inhibitor. It's a formal complex with the E2A. And this complex, the E2A is important for driving T cell differentiation. So when you have ID3 there, it suppresses the T cell differentiation, therefore it promotes memory formation and memory recall response. And also a marker of PD-1 positive exhaustion T cell precursor cell population. It has an important role in CD4 T cells. But whether ID3 is important for tissue infiltrating polyfunctional CD4 Th1 cells in the GVHD is unclear. So in one of my postdocs, she worked with me for nearly 10 years now. She found that ID3 play important role in reduce the chromatin accessibility to the transcription factor, including these transcription factor, which are important for the promoting the expression of PD-1, PD-L1, CTL4, TIM3, and the CD9, CD39, which are all immunosuppressive inhibitor or receptor on the surface of T cells. If you don't have ID3, these molecules will be upregulated and increase the susceptibility of these cells to the immunosuppressive molecule ligand, such as PD-L1 and so on. So on the other hand, ID3 has an intrinsic role in reducing the T cell dysfunction differentiation and while promoting the generation of long-lasting TCF1 positive cells. So therefore, if you delete ID3, you will selectively reduce the GVHD in the non-hematopoietic tissue. But something question still there. We do not know how easy, whether ID3 act as an epigenetic regulator. If yes, how? And also, how is ID3 function maintained in tissue infiltrating GVHD T cells? What the non-GVHD tissue, non-hematopoietic tissue producing some factor regulating the ID3 and so on. So one of my postdocs, including in addition to the Yin, working on this part. So I hope sometime later we can update you the progress of this research part. So therefore, EZH2 has a multiple function in the T cell immunity. It's involved in the regulation of Treg, TFH, follicular T cell, Th2, basically it suppresses Th2. It represses the terminal differentiation and promote the membrane precursor cell formation and so on. So this is an important paper for the chronic GVHD study, which is published by Dr. Blazer's group. Now, so the question is that we ask, why you don't have EZH2 antigen activated T cell will die? What is the molecular mechanism there? Because if T cell die, the story ends. You cannot further study what is the biology of EZH2 in T cell. So we spent nearly 6-7 years to figure out what's going on. At the beginning, we delete one gene by one gene. We delete nearly 10 to 11 transcription factor downstream of the EZH2. We do not see good response, so therefore it's not that one single molecule there. So we did RNA-seq again. We asked what kind of a gene changed, which could be, and it could have a broad impact on T cell. We found that calcium. If you don't have a EZH2 there, calcium signaling activity is significantly activated. And then we use the calcium assay, the cytosolic influx assay, which is done by Jonathan Sobolov's group. He recently moved to cancers. So he found that in the absence of EZH2, calcium response is significantly increased. So this indicates that the EZH2 may modulate calcium signaling. So this is a calcium release. So normally when T cells are activated, now I have a PLC gamma activated. Now you open the channel ITPL3 channel, protein channel. Now the calcium released to cytosolic and activate calcineurin. Now you have MFAT activation. That's how we use the calcineurin inhibitor to suppress the GVHD. It can also release to the mitochondria and cause the increase the MPTP. So you have like a pool formation. Now you induce cell death. Of course, that's also important for mitochondrial metabolism. So Craig Bystoff is an expert of metabolism parts. Now, once calcium is released, now STEAM1 in the UR is activated. Now migrate to the cytoplasm, form a complex with the ORAL1. Now this channel opened, and you have calcium influx. So therefore, we ask, say, may be easy to regulate the STIM1 channel or regulate this kind of T cell signaling channel, calcium signaling channel. Therefore, you have calcium signaling altered. So we use a STIM1 knockout mice, and we found that the STIM1 knockout mice, knockout T cell, had a significantly reduced capacity to induce a GVHD. And we overlap the gene altered in STIM1 knockout GVHD T cell with the easy to knock out reactive T cell. We found around about 41% of the gene overlapped. And among these overlapped gene, they are cell cycle regulator. They are here is that T cell differentiation regulator, and the senescence pathway regulator, and so on. And you just see the opposite. So when you have a STIM1 knockout, you see these gene activity of the pathway are enhanced, but these gene pathway in easy to knockout cells are reduced. You have a group of genes, also expression totally opposite. So you see in the absence of STIM1, you see these genes upregulated. But in the absence of EZ2, these genes are downregulated. So it's just totally opposite. And here you see some of the genes like overlapped. So this indicates that either EZ2 regulates STIM1 or STIM1 modulates EZ2. And STIM1 EZ2 knockout cells may die because you have enhanced calcium influx, the calcium activity there. So to test this hypothesis, we generated the EZ2 STIM1 double knockout mice. We had the trouble at the beginning because if you double knock out these two molecules during the thymopoiesis development stage, the whole thymopoiesis is kind of disrupted. So we had to use the mature T cell, express this flux gene, and we deliver TAT3 protein and selectively deleted both protein in this mature T cell. And then we measure the activation and the survival of these EZ2 knockout cells versus, and also double knockout, we name it the ES knockout. Now you see significantly improve the survival capacity in both the CD4 and the CD8 T cells. This is surprising to us. When you block the calcium signals, now you have a rescue. So we term that a synthetic rescue. So it means like you delete the STIM1, now you reduce the calcium signaling. Now you can reduce the rescue the EZ2 the deficient T cells. And then when you put these cells in vivo, now you rescue the capacity of these EZ2 knockout cells to induce a GVHD. We did a lot of other assay about the in vivo T cell response assays, so I will not show here. You can also rescue the EZ2 knockout T cell if you deplete STIM1. So this means like EZ2 really is important for regulation, replacing the calcium signal. I cannot say replacing, modulating. We hypothesize that one, it may be replacing the STIM1 or oral expression, or maybe downstream pathways such as RTPR system or the other calcium signaling part. So we measure the STIM1 expression, STIM1 oral, and you don't see that much change, but you do see a significant increase expression of gene coding PLC gamma 1, PLC gamma 2, which is important for mediating the T cell signaling to activate this ITPR channel to release the calcium. But one thing is very interesting and surprising to us, you do see upregulation of RTPR1 and 2 in EZ2 knockout cells. We retrieved the data from Restifo's group about the H3K27 chip assay, and we found the ITPR2 present, the H3K27, which is an indicator of our EZ2 functionality, present at the promoter region, and also present at the promoter region of the ITPR1, but not ITPR3. So, and then we start the crossover ITP1 knockout to EZH2 knockout, ITPR2 knockout to EZH2 knockout, and so on. So we found that ITPR2 and the EZH2 double knockout can rescue the EZH2 knockout in the context of inducing the GVHD. Now, the kind of a caveat here is that polyfunctional T cell. So in the GVHD model, you have alloantigen specific T cell. You also have a T cell undergoing non-alloreactive T cell undergoing an expansion proliferation. So we want to understand that the specific reaction specific effect of T cell signaling in the ITPR2 and EZH2 knockout. So we generate the CAR T cell because now you have a CD19 antigen specifically activate those CD19 CAR. So we put the CD19, human CD19 CAR into a retroviral vector, and we generate the CD19, human CD19 expressing C1498 cells, which is leukemia, AML leukemia cells. And we put them in vivo. Now you see, if you have an EZH2 knockout, you cannot eliminate tumor. Now, if you have an EZH2 and the ITPR2 double knockout, now you can rescue the capacity and restore the capacity to reduce, to eliminate leukemia. So this indicates that EZH2 repression of ITPR2 is very important for regulating this calcium signaling parts. So we also measure test the hypothesis whether calcium may affect the EZH2 because we see like a kind of interdependent, co-regulation. So we use a tacrolimus, which is a calcineurin inhibitor, and treat the CAR T cells, and we see the increase the H3K27, and we also see reduce the EZ2 silenced gene here in relatively increased ID3 expression in these CAR T cells. So this indicates that if you inhibit the calcium signaling in CAR T cell, you can enhance the EZ2 functionality. And indeed, when we use the CAR T model, if you infuse the tacrolimus treated CAR T cell, you see relatively enhanced anti-tumor activity, indicating this EZ2 activity is really enhanced in these CAR T cells. And then you see these cells, you see the reduce the exhaustion phenotype cells. So surprisingly, again, when we had these data, and I started writing, and of course, I found Bruce Brazer's paper, and this published in 2020 in the Blood, and his group found that if you boost the calcium and cytosolic calcium response by removing the inositol kinase B, which is ITPR inhibitor, now you have a reduced GVHD, exactly consistent, with our finding. So I was so happy that, our hypothesis be, very much, well supported by, Bruce, the finding. That's why I say, he's a library. If you check whatever, you can find something, and it can corroborate your findings, so on. So here is a brief summary. So basically EZ2 modulating ITPR expression, and then EZ2 may also regulating the gene activated by calcium signaling, and therefore modulate T cell response. If you remove the calcium signaling, even if you have a downregulated EZ2 functionality, that could be fine. You can synthetically rescue the EZ2 function, and then enhance the T cell response or restore the T cell response. So now I switch the gear a little bit to the last part about the EZ2 and the anti-tumor immunity. So this is a brief summary showing how T cell response and the persistence of T cells response is required for eliminating tumor. So first of all, step one, you need to activate the T cell and generate a large amount of the T cell. And these T cells have to infiltrate into the tumor sites and expand there. And then they execute the function to eliminate that tumor. Any step missing, any kind of dysfunctional change among these three steps, well, you will have kind of failure to eliminate tumor, because for the effective complete elimination of the tumor, you need like up to like months and years, to maintain that activity. So this is indeed, kind of, observed in the CAR T cell field. So CAR T cell has, transformed the cancer immunotherapy, in particularly in the treatment of B-cell malignancies. But, unfortunately, still you have 50% of the patients with a B-cell malignancy. They experience a non-relapse in the disease, non-response in the disease relapse. The efficacy of CAR T cell in solid tumors still not well established yet. So the CAR T cell dysfunction is one of the major causes of the failure of CAR T cell therapy. That's including CAR T cell exhaustion, loss of stamina and the memory, and it becomes silencing, and also they may lose the capacity to traffic into the tumor site. So the several epigenetic regulators have been demonstrated to replace the CAR T cell functionality, or to drive the CAR T cell exhaustion, memory loss, and the senescence. It's very difficult to separate senescence from memory loss and exhaustion, because you have an overlap the phenotype, and the senescence itself involves the memory loss and the exhaustion and so on. So, so TAT2, which was the first published by Carl Jung's group in Nature a couple of years ago, there was a CAR T cell therapy patient. That's a CLL, patient. They found that the CAR T cell, the proliferate persist over many years, all carry kind of insertion mutation, that is TAT2. And the subsequent the experimental study showing if you delete the TAT2, you can have enhanced expansion, reduced exhaustion of the CAR T cell. Therefore, TAT2 is important for driving the exhaustion, memory loss, and the senescence. And so the other group also found that DMT3A by Young Brother group in St. Judy. And this is by Michelle Salin's group, SUV39H1, which catalyzes H3K9. So these are all like play a role like a suppressor. So our question is that, do you have an activator? Does the epigenetic regulator exist to promote and maintain CAR T cell proliferation capacity while limiting exhaustion or senescence? So bearing this question, we try to set up a system to understand and identify what kind of molecule there. So for the T cell, proliferation, exhaustion, and the memory loss always coupled together. So it's very much like a man, you cannot have this cake, but you want to eat this cake. It's a balance. So what is the balancer here? We retrieved the data from Carl Jung and Dr. Fran's group, Fran's from Yale. So they had a clinical study with 82 patients. So we retrieved the data. We select the samples with a CD3 positive CD3 expression CAR T, and that are activated before infusion. And there are 6 samples from healthy donor, 25 samples from responder, 6 samples from non-responder. So these data are pulled together, and we look at what's going on there. This is a single cell seq analysis for the CD8 population. You see several clusters, and one of the cluster, two clusters, cluster 2 and 7, they're highly proliferative, expressing MK60, MKI67, which is an indicator of proliferation cells. And we use a cytotrace which is a software for identify less differentiated cells with stemness. So you see cluster 2, which is a proliferating cell. You have really highest score of a cytotrace, means like they are less differentiated. So which is surprising to us. And we further analyze what kind of gene there. So we separate into several categories: memory gene, effector gene, exhaustion gene, and a proliferating gene. So cluster 2 is a highly proliferated cells. They are effector-like cells because they express interferon gamma, the CSF2, that's the IO2. You have another proliferating cell population. They express memory markers. So you have a two population proliferation, undergoing proliferation. One is a memory phenotype cells, one is effector phenotype cells, but they're all highly proliferative cells. So if you look at this cluster 2, you have a typical cell cycle regulator gene there, but one cluster of the gene is very important for us, that is telomere maintenance. So during T cell proliferation, you need to maintain the telomere. And if you have a shortened telomere or you have mass up the telomere, you will have a kind of cell death and senescence and everything ongoing. Now, in addition to these category genes, if we extract all of these genes or look at the representative gene, what are they? So they are all genes related to and very important for embryonic stem cell, neural stem cell proliferation, survival, and self-renew. So, and we also look into the epigenetic regulator. So you see now PRC2, EZH2 complex there. You also see DMT3A and SUV39H1 there. So it looks like in this particular population, you have a two category of gene there, maintain the balance. So one is a TAT, DMT3, which drive the exhaustion and memory loss. And now another one is EZH2 PRC2. What is the role of this one? So we start to search the literature. Two papers come up. So only this year, we actually spent 6-7 years working on this project. Now, only this year, people published a paper in Cancer Cell. This is from Cornell University. They found that if you inhibit the tumor EZH2, you can improve the T cell anti-tumor immunity. This is a paper from Penn, Mark Aurelius group. They found that if you inhibit B-cell malignant bearing mice with the EZH2 inhibitor, you can also enhance the anti-tumor immunity. But none of these papers fully examine the precise role of the EZH2 human CAR T cells. So this is a paper published by Christian Macau's group in Science a couple of years ago. They found that tonic signaling induced the exhausted T cell express a kind of a gene, exhaustion gene, and somehow they are replaced by a mark, they are marked by H3K27. Again, the precise role of EZH2 in these CAR T cells was not examined. So we built up a system using the CRISPR knockout. So we delete the EZH2 in human CAR T cells, and we found that you suppress the T cell proliferation. And we use the EZH2 inhibitor. This is DS302, that's from Daiichi Sankyo. So early on, you don't see that dramatic impact in the 4 days, which is exactly like what we saw in the mouse model system within 3-4 days. T cell without EZH2 could be okay, but after 4 days, you see collapse. Now you see day 6, you see significant reduced expansion. By day 8, you see you have around a 10-fold differences. So those papers who examine the EZH2 in T cell, just roughly, they measure the short term. For epigenetic regulator, you really want to monitor the impact of this inhibitor a long time. It's not just a short term. That's so-called epigenetic inheritable imprinting and the change in program. Now, if you look at the anti-tumor immunity, it's gone. it's basically you don't have anti-tumor immunity. Now, if you use the CAR T cell, if you simply treat with the EZH2 inhibitor for 6 days, and then you put them back into the mice, do they, retain the capacity, or can the suppressive be reversed? The answer is no. So we are preparing the manuscript. So we clearly show the EZH2 is critical for the CAR T cell elimination of tumor. Now, what is the mechanism? So you do see reduced number for CD4. This is all human cells in the NSG mice. And here you see the TCMA reduced, the memory response reduced, so that's not surprising. But we do see TMRA, which is terminally differentiated, a senescent T cell, significantly increase. You have up to 80 or 90% of them are TMRA cells. That's indicating either terminal differentiation or senescence. So you do have a terminal exhausted cell phenotype there, but not that much later stage for CD8 cells. You don't have a big differences, maybe because here EZH2 functionality already dropped and so on. So to address at the molecular level, whether this really kind of senescence issue and so on, we did cut and run first. We look at the EZH2 binding gene and the H3K27 marker gene. You have a solo gene, means like you only have EZH2 there. Now, these EZH2 only binding gene, you have a senescence in addition to cell cycle and the other T cell response gene. And we did RNA-seq, and we found these signaling pathway. In particularly STAT3, which is indicate also a senescence pathway, is activated. And you see cell cycle activator, including TOP2A, CCNB1, and CCNB2, CDK1, significantly reduced. Other genes, including the enhancing molecule, is increased in EZH2 knockout cell. So this part exactly consistent with other people, data showing the, EZH2 silence gene. But unlike this, EZH2's chronic gene activity, EZH2 has a non-clinical function, which is activate cell cycle activators. So we use the chip track to monitor the EZH2 binding at the promoter region of this TOP2A. And these are the EZH2 and the K27 and dual presence at the promoter region of enhancing molecule. And this is transcription factor regulating the exhaustion and differentiation. So to further understand whether verify EZH2 inhibition leading to the senescence, we monitor four different things. One, do have increased the cell cycle inhibitor, which drive the cell senescence? Yes, you see P21, P16, P15 all increase. That is an inhibitor drive the stem cell senescence and so on. And we look at the telomere length. If you have an EZH2 inhibitor there, you see telomere reduced. That's why when you put these cells back into the animal model, they cannot be reversed because once it's shortened, you cannot reverse that. And we look at the cell cycle. So this green is a G2M. You see about a tenfold reduce if you culture them with the EZH2 inhibitor. Beta-gal is one important indicator of a cell senescence. You see significant increase the activity of beta-gal. So this indicates that EZH2 really is important for limiting, restricting the senescence. And what's the role of TOP2A? So if you inhibit EZH2, you see TOP2A reduced. Now, if you inhibit TOP2A, you see T cell expansion, proliferation is reduced. You also see telomere shortened. If you look at the EZH2, now if you have a TOP2A inhibitor there, you see EZH2 function is reduced. If you recover these TOP2A treated cells, put them back into the leukemia mice, these CAR T cells lost the capacity to eliminate tumor. That therefore, once you have a top-2A treatment, you induce irreversible change. That explains why some kind of DNA, inhibitor chemotherapy agents may cause really kind of T cell dysfunction, and that could be kind of trouble if you use a long-term high dose. And then we discover a self-reinforcing regulator loop, where the EZH2 activate this cell cycle activator, and then top2A can activate EZH2 expression. So this self-regulatory loop is very important in sustaining the proliferation and also limiting the senescence in proliferating cells. Unfortunately, if you have a high tumor burden, or you have like a long-term prolonged exposure CAR T cell to tumor, EZH2 functionality is disrupted. So we set up a mouse model. You have a high tumor burden, which means like you get a low number of CAR T cell, you have a relative high tumor burden. Or versus you get high number of CAR T cell, you relatively have lower tumor burden. Now you see high tumor burden, you see the persistence of leukemia there. Now you see CAR T cell significantly reduce the EZH2 expression, EZH2 functionality. We can use flow cytometry analysis to monitor that. And we set up another culture system using the high tumor burden culture. You have 1 to 9, so it means that 1 CAR T cell versus 9 the tumor. And you also see induced CAR T cell dysfunction. You lost the CAR T cell in the culture. You also see significant reduced the EZH2 in both the CD4 and CD8 T cells. And this is antigen stimulation relevant. If you delete the CD19 antigen in this Raja cell, now you see the EZH2 expression is not impaired. So this is a core signaling mediated degradation of the EZH2. So because time concerns, so wrap up here. So continuous activation disrupt the EZH2 in the CAR T cell, and inhibiting EZH2 in CAR T cell results in impair the proliferation and enhance the senescence. So this indicates that in clinic, if you gave the EZH2 inhibitor a long time, you probably have a trouble. So another one, we found that EZH2 in the top2A form a self-reinforcing regulator loop. So this loop is important for dissociating the proliferation from senescence. So the next question is to understand, can CAR T cell be pre-programmed to render them EZH2 dependent superior anti-tumor activity? Means like enable these CAR T cell resist tumor, prolonged tumor stimulation and disruption of CAR T cell. So this work is on the way. We hope to finish the work very soon, within 1 or 2 months. So overall, today we discussed about the role of EZH2. It has a really multiple role there. So we will focus on one is how EZH2 be used as a target to modulate tissues specific GVHD. So Dr. Blazer's group also discovered that this EZH2 pathway can be used for modulating the chronic GVHD, which is a major issue in the clinic. And we also found that EZH2 and the calcium coordinate the signal to induce the productive factor response. If you don't have EZH2, T cell may die because the calcium influx is significantly increased. T cell undergoing apoptosis, and also the terminal differentiation senescence. And finally, we found that using human CAR T cell, EZH2 is important as a master regulator, and coupling the different proliferation and the senescence, which is important for improving the efficacy of T cell immunotherapy. So these work are done by over a period of many years by Ying, Mimi, and Ron, she's the person working on on T cell HSP90 also. I have three groups work for to assist us bioinformatic analysis: Corey Institute, John Pierce group, the Fox Chase group, and also my long-term collaborator in China. Without their help, we could not have this kind of accomplishing the EZH2 study. I will stop here. Thank you.
I have microphones for anyone with questions.
Congratulations, you had a very comprehensive talk. I'm trying to understand the role of EZH2. You have different role in CAR T cells in terms of having the anti-tumor effect in alloreactive T cells. So how would you, can't really do this in vivo at this point, but if you did, you'd have a difference between inhibiting GVHD, but also changing the CAR T cell anti-leukemia effect. How does that work?
So let's say this way. So one is the mouse model part, and one is the human CAR T cell parts. Mouse T cell and the human T cell might differ a little bit. For example, in the mouse T cell, easy to directly activate ID3, but in human T cell doesn't. A human T cell easy to not show relative increased expression of ID3. This is one thing different. Second one is the oldest study in our studies and the other groups. They did not pay attention to the senescence part. In particular from the Suke's group, so she studied the EZH2 knockout, and her conclusion is that EZH2 promote memory cells, and that's totally opposite to what we observed. So Luca Gattonini and I, we called together, we discussed, we could not figure out what's going on there. It could be like, the model issue or, time kinetic issue. But one thing is very clear. The only impaired old expansion and also increased apoptosis may mask the phenotype, and you lost the opportunity to look what's going on there. For example, in the mouse T cell, 7 days after the transplantation, GVHD, you see significantly impaired expansion of T cells. Therefore, you cannot track those cells, what's going on there. Unlike that, for human CAR T cell, somehow they can survive a little bit longer in NSG mice. And then that allows you to monitor the senescence.
So I just, 2 short questions. That for the epigenetic memory, as the cells divide, how does that work, that both daughter populations are imprinted? What stays there? Is it other regulators that are altered?
Yeah, definitely. Yes. It's a very interesting question, Bruce. I even cannot answer this question well now, because when we knock out EZH2 in human CAR T cells, you see relatively increased TCF7, you see relatively increased ID3. Therefore, it is not that the impact on the memory gene program, but we do see significantly activated AKT PI3 kinase pathway, SOC kinase pathway. Therefore, somehow the balance between the AKT pathway and this memory gene program somehow kind of messed up. This is one thing. Second one is that my postdoc's idea. I've been schooled by the memory gene program. And you should say, I don't agree with this idea. EZH2, knockout, reduce the memory in a way, kind of simple way. And you should say that you see the suppressed expansion. And this kind of suppression of the expansion means a lot. You do not have a pore size be expanded, including effect, including memory. Therefore, you lost the capacity to undergo self-renewing of the memory, although you have those gene expression there. And then where these cells go? I mean, if they don't expand, then why is that they survive? No, that they die. That's calcium. And another way is that they differentiate, skewed towards the senescence, and then they stop.
Thank you for that excellent talk. And just wondering how you see EZH2 calcium flux and downstream effects interacting with TCR signal strength and sort of high versus low affinity T cells.
We do not check it. It's a very important question. Actually, we've been stuck with the reviews question now. So we need to figure it out. That's in the revision. Yeah. So my guess, I only can guess because we don't have a low affinity TCR signaling system there. But my guess, if you have lower affinity of T cell signaling, TCR signaling, EZH2 might not that important in terms of the regulating the survival, because we found that under homeostatic condition, which means like you only have self-antigen, now you delete EZH2, it doesn't matter. They can still survive and proliferate. Although they undergo long-term, they undergo differentiation, become senescent cells. We do see that in aged mice, you do see that, but in short-term window, you don't see that one. So that's the only answer I can have.
All right, we'll take one last question here with Jaime.
Yeah, this is related. So I think it was maybe about 20, 25 years ago, Kim Bottomley showed that when you have signal 1 without signal 2 or a low affinity signal, you get permanent energy, right? And
yeah.
I think in looking at your data, one of the questions is, what is creating the irreversible epigenetic change? It seems to me that these epigenetic changes should be really dynamic, and they are up to a point, and then they seem to get locked. So what do you think is the difference or the signal strength that's required to lock the epigenetic state into irreversible energy or death or senescence versus being able to go back and reactivate?
So again, this is a great question. It can be like $1 million of for work. So what I can answer at this moment is that one is the balance between cell cycle activating inhibitor changed, but how that change be maintained, we do not know at this moment. So we did the kind of a very rough assay. We use a CFAC, CVT assay. We can monitor during the dividing, is easy to maintain, the top 2 maintained. And yes, in that particular population, we have maintained over several divisions, therefore it's maintained. But how is it maintained? Is that because other pattern of like a demethylation pattern, demethylation and so on? We're doing that. We're using nanopore to sequence to try to identify, find the solution. But we know that Dr. Bystof's the idea about metabolism is there, and once hit, it lasts forever, and that could be like relevant to the more likely DNA methylation pattern. We also found that the paper, the data was not published. They say if you have EZH2 knockout cells, CD8, we use a mining energy H60 to track that, and also PML. 160 days or 200 days after, you recover those knockout cells. They are still knockout. The small population still survive. They are anergic. They don't respond to the antigen stimulation. We have the central member like a phenotype there. We never published that data. I wish people in the field can be more interested in understanding what's going on there. But this is a really important question.
All right, let's say thank you one more time to Dr. Zhang.
Thank you.